It's okay with me. All right, today we're going to discuss agents affecting water conservation. Uh, First, a little bit of an overview. Obviously, maintaining consistent body fluid osmolality is vital, and everybody remembers osmolality from physics and stuff like that, right? I'm not going to go into the lengthy explanation. I asked my friend Taberge if he explained osmolality to you guys, and he said no, and I'm like, he's the chemist, but I don't know. Essentially, for our purposes, I think you can mainly think of it how much water is in the blood. It's not how much blood there is; it's how much water is in the blood. Is kind of at least a useful way to think of it. Okay, arginine vasopressin, also known as vasopressin and antidiuretic hormone, is the main hormone that's involved in water balance regulation in the body. It's a nonapeptide, which means it's just 9 amino acids stuck together. And treatments for water balance disorders in the body often involve vasopressin. And there's also a number of disease states, as you would guess, with something that's involved in regulating water in the body. A number of disease states can affect vasopressin actions, particularly things like diabetes, stuff that affects the kidney a lot. Vasopressin is highly evolutionarily conserved, so it's a molecule involved in water conservation in a lot of different animals, vertebrates, and also invertebrates. Once again, this vasopressin is there to mediate the conservation of water. As you can imagine, water is a fairly precious resource in the Western world. We've got it kind of easy, where we have water readily available, but historically, people would go through times of drought and things like that. So, water is not always there and available. So, the body developed ways to conserve it. Okay. In terms of where vasopressin is made, it's made in the hypothalamus, in the supraoptic and paraventricular nucleus of the hypothalamus, and it's released from the posterior pituitary. And you guys remember the posterior pituitary is the part of the pituitary that's actually neural tissue still. It's just basically a projection of the brain, as opposed to the anterior pituitary, which is attached to the brain by blood vessels. So vasopressin made in the brain, secreted from the posterior pituitary. This slide just jumps right into what does vasopressin do, or it actually does several things. But this slide shows what vasopressin's action on water reabsorption in the kidney, and this is actually supposed to be this is the collecting duct of the kidney. So what we see on this side of the slide is the antidiuretic hormone or vasopressin is not present. So, and what we have here is these big arrows are salt reabsorption, and then over here we have urea and water reabsorption. So with vasopressin absent, we'll still get salt reabsorption, etc. Sodium reabsorption. When vasopressin is present, what happens is these pores open up and water is reabsorbed. Okay, so it increases water reabsorption. And also, another effect of it is urea gets reabsorbed as well. So an increase in urea reabsorption. Okay. Vasopressin is released in response to several conditions, including increased plasma osmolality, hypovolemia, and hypotension. Okay, so if you don't have enough water in the blood, vasopressin is going to be released. Vasopressin has many actions at the level of the kidney. Those receptors where you get the pores that cause the pores to open up, those receptors are found in the renal collecting duct. And so what happens is the vasopressin receptor activation causes that increase in permeability of kidney cells to water, and that just allows the water, once again, to move out and be reabsorbed. Vasopressin's other action, when you think of vasopressin, you kind of think it has two main actions. They're separate from each other, actually, to a certain extent. It's also a potent vasoconstrictor. And it also has some other peripheral effects that include causing the release of coagulation factors from the vascular endothelium and increasing platelet aggregation. Okay, and then, like a lot of other agents that are made in the hypothalamus, other hypothalamic peptides, it can also act as a neurotransmitter, and it's involved as a neurotransmitter largely in things like water conservation, affecting areas of the brain that deal with that. It also affects adrenocorticotropin hormone release from the anterior pituitary. If you remember, ACTH is part of the HPA axis where cortisol is released. So vasopressin can cause ACTH release, and it's involved to a certain extent in regulation of cardiovascular function and body temperature. Okay, a little bit more about vasopressin, more specifics in terms of synthesis, storage, and transport. Like oxytocin, it's synthesized once again in the supraoptic and paraventricular nuclei of the hypothalamus. Those axons project down to the posterior pituitary where it gets released. It starts out as a pre-pro hormone. As in fact, you've probably been over a few pre-pro hormones before when you went into your endocrine stuff. So all that means is that vasopressin, when the vasopressin gene is made, so our other genes are transcribed as well, and they're all stuck together in one big long prohormone that then gets cleaved up. So the ones that are made with vasopressin are neurophysin and copeptin. Okay. It gets transported into secretory granules in the cell body where it's being made in the brain, and then those secretory granules travel to the site, from the site of synthesis to secretory terminals in about 30 minutes or so relatively rapidly. It's also synthesized outside the CNS. I'm not going to really get into that, but vasopressin is made in the heart and in the adrenal gland as well. And you see that a lot with these hypothalamic peptides. They're made in the skin as well. Things like ACTH is made in the skin, and you can actually do something like rub ACTH on yourself and stimulate hair growth, things like that. So these things have different actions in different places. This slide shows a simplistic version of vasopressin synthesis. Bless you. So up here we have the vasopressin precursor, the pre-pro hormone. So we have vasopressin, neurophysin, and copeptin all together. They get shuttled over to the endoplasmic reticulum where they're cleaved to a certain extent. A signal peptide at least starts the cleavage. Then at the level of the Golgi, they're broken down into vasopressin, neurophysin, and copeptin, and then ththose guys are in the secretory vesicles that get released. This is the same thing. All Goodman and Gilman. I'm not going to expect you guys to memorize the endopeptidases or anything like that that cleave vasopressin. You should probably be aware they exist, though. To my knowledge, there's no drugs that go after them at this point, so I'm not going to worry about that too much in terms of specifics, but I think you should realize they exist in case down the road any drug does get invented that alters vasopressin synthesis. This slide shows the sites of synthesis and release of vasopressin. It's kind of simplistic, but up here is supposed to be the hypothalamus, and this would be the paraventricular nucleus right here, and here's the supraoptic nucleus. And so these neurons that make vasopressin send these long projections down to the posterior pituitary, where it gets released in response to changes in osmolality and hypovolemia, etc. Once again, vasopressin is released in response to increased plasma osmolality. Please remember that, increased plasma osmolality. Don't miss it on a test question just because you ran over it real fast. And once again, released in response to hypovolemia, so just a loss of volume, plasma volume, hypotension, and also in response to pain, nausea, and things like hypoxia. So a real simple way to think of it is, and it's true, is vasopressin is released in response to stressors, homeostatic challenges. The osmolality threshold, you don't need to memorize this for the secretion of vasopressin, is 280 milliolemolars per kilogram. You don't need to worry about that. But basically, what you should be aware of is about a 2% increase in osmolality causes a 2 to 3-fold increase in plasma vasopressin. And vasopressin is going to work to rapidly cause the reabsorption of water and lower osmolality again. And what that does in terms of your physical being is it allows for longer periods of where you're not thirsty. You don't need to take in water, which, can be handy when you're living in a desert and things like that, and you want to survive. There's several CNS areas that are involved in the osmotic stimulation of vasopressin secretion, and collectively these guys are referred to as osmoreceptive complex. And these include the supraoptic nucleus and the paraventricular nucleus, subcortical organ, the organum vasculosum of the lamina terminalis, and the medial preoptic nucleus. So again, you don't really need to memorize those, but you should know that the areas of the brain that are involved in sensing changes in osmolality are called the osmoreceptive complex, and that what they do is they sense changes in osmolality. And you notice how I can actually pronounce brain things, but not drug things. So I think that I'm a neuroscientist, right? Okay, I can take pride in something. Okay. So this slide, in a kind of a simplistic form, shows the regulation of vasopressin secretion. So, and here they have it actually as a nucleus tract solitaris. But what happens is in the brain and also in the liver, there's osmoreceptors, and when they're stimulated, they send signals up afferent neurons up to the brainstem, and then stimulate different areas of the brain. Here we have the subcortical organ, the medial preoptic nucleus, the organum vasculosum, lateral terminalis. These guys all work in collusion to ultimately stimulate the release of vasopressin, once again, when these osmoreceptors are activated. Okay. In the brain, there's a molecule called aquaporin-4. It's a water-selective channel on cells that are actually involved in regulating osmolality. So that's actually found on, it's interesting, it's found on glial cells and ependymal cells, not on neurons. And so what that really means is there's a glial neuron interaction in the regulation of vasopressin release and osmolality. So these pores, what they're doing is they're allowing water to come in and, in these osmoreceptors. And so, that's how the brain is sensing water levels, essentially, in addition to getting signals from the periphery. Okay, and once again, those peripheral signals are these hepatic portal osmoreceptors, and the way those get activated is actually by salt. And once again, when those guys are activated, the liver, acetylcholine, afferent nerves that use acetylcholine as a neurotransmitter send the signal up to the brainstem, and then that signal gets to the organum, vasculosum, lamina terminalis, etc., paraventricular nucleus, supraoptic nucleus, and what happens at the end of the day is you get vasopressin release. This slide just shows aquaporin structure. So once again, aquaporin 4 is found in the brain, and essentially all it is a pore that they'll be hanging out in the plasma and it'll be inserted into the membrane of the cell when it's stimulated to let that water in. Okay, hypovolemia, so just loss of total volume, and hypotension actually induce vasopressin by a different route, so it's not via these osmoreceptors, and it doesn't interfere with vasopressin's osmolality regulation. So these are actually, they're two different things, right? Okay, and the regulation uses different neuronal pathways than osmolality, and so small changes in blood pressure don't cause, don't cause vasopressin release. So what we're talking, I guess the way to think about this is there's a difference between changes in blood pressure that you're going to see with hypotension, things like that, the way that stimulates vasopressin release, and changes in osmolality. Small changes in osmolality are going to cause some decent release of vasopressin. Small changes in blood pressure don't. You need a fairly big change in blood pressure to get vasopressin released. When it does get released, following that, and it acts as a vasoconstrictor, and it's kind of an emergency system. Vasopressin gets released in large amounts when there's a big change in blood pressure, and you get this vasoconstriction and stuff like that, and that helps prevent cardiovascular collapse when you're bleeding a lot. So, you're walking down the street and fall over and fall on a stake or something, and you're bleeding profusely, you're going to get this big dump of vasopressin that happens, and your veins are going to constrict, etc., and that'll help keep you from losing too much blood. Whereas if you're walking down the street and you nick yourself a little tiny bit and you lose little teensy bits of blood, that's not going to cause it. Okay. Vasopressin release can also be stimulated or inhibited by hormones and neurotransmitters. Some agents that stimulate vasopressin release include acetylcholine via nicotinic receptors, dopamine via D1 and D2 receptors, and then also glutamine, aspartate, cholecystokinin, neuropeptide Y, etc. I guess the key thing is if you have a drug like nicotine that affects the cholinergic system, you have a chance of affecting vasopressin release. If you have a drug like an antipsychotic, which is blocking D2 receptors, it can have an effect of altering vasopressin release. Agents that inhibit vasopressin release include GABA. So that would also mean, by extension, something like alcohol that enhances GABA function is going to inhibit vasopressin release. Atrial natriuretic peptide and opioids. So those all inhibit, the others stimulate. Okay. Pharmacological agents, which I sort of alluded to a second ago, also alter vasopressin release, and they do so by actions on receptors or by changing plasma osmolality. These agents can, on their own, change plasma osmolality. I have a laundry list of agents that can stimulate vasopressin secretion. I think the big one would, you'd probably want to know at least nicotine. High morphine doses can, and so can antidepressants. They can actually cause stimulation. So, and you think of it, antidepressants, some antidepressants actually increase dopamine, right? So, and if dopamine receptors are involved in vasopressin release, stimulating it, follows through. The antidepressants can actually cause it to be released. Agents that inhibit vasopressin release, once again, alcohol can do it. phenytoin could do it. Low morphine doses, glucocorticoids, and then antipsychotics, things like that. When you're blocking dopamine receptors and not instead of stimulating them. In terms of how the body responds to vasopressin, it's of course going to be through vasopressin receptors. They're all G protein coupled receptors. 3 types exist: V1A, V1B, and V2. You want to keep these straight, at least until you take tests on them. So the V1 receptors are coupled to GQ, and also will stimulate the opening of a calcium channel, and they can activate eicosanoid synthesis. Which is, remember, vasopressin can have an effect on platelet aggregation, things like that. V2 receptors coupled to GS. So, just as a simple question, knowing what you know about vasoconstriction, right? Vasopressin causes vasoconstriction. Which of these receptors are going to be on vascular smooth muscle? Should be able to figure out from just that slide. TikTok. This is the closest I get to a crossword puzzle. What's that? Oh, the question is, if knowing what you know, vasopressin causes vasoconstriction, right? So, knowing what you know about what causes vasoconstriction, previous lectures, what type of receptor causes vasoconstriction, which one of these V1 receptors, which couple to GQ, or V2 receptors, which couple to GS, which one is going to cause which one is going to be found on the vasculature to cause vasoconstriction? V1, right? So the calcium that's released, calcium causing constriction. If V2 receptors were found on the vasculature, they couple to GS proteins. And what happens when you stimulate GS proteins on the vasculature, you get relaxation, right? Just the question was trying to get a feel for G proteins and what they're doing. Okay. In terms of these vasopressin receptors, V1 is the most widely expressed. It's the one, once again, that's found in vascular smooth muscle, plus other tissues, including the CNS. V2 is found in the anterior pituitary, V1B is found in the anterior pituitary, also the CNS and pancreas, and then the adrenal medulla. So V1B would be the one that's involved in ACTH secretion. Okay, V2 is the one that's found in the kidney. So it's found mainly in principal cells of the renal collecting duct, okay, and also on epithelial cells of the thick ascending loop of Henle. And then it can also be found on some vascular endothelial cells. Okay, so pretty easy to try, if you want to keep these straight, V1, vasculature, constriction, so you can, I would hope, be able to easily remember they're going to constriction, so they coupled to GQ and V2, think of mainly at the kidney. Okay, V1 activation, once again, results in increased intracellular calcium levels. So with that, you get contraction of vascular smooth muscle. You also get at the level of the kidney, when these V1 receptors are found there, contraction of mesangial cells in the glomerulus, and you can also get prostaglandin synthesis. This slide shows, in simplistic form, V1 receptor activation. So here is your V1 receptor, and when it's activated, you're going to stimulate phospholipase C activity, increase inositol phosphate. Ultimately, you're going to get a release of calcium from intracellular stores. That calcium is going to then have other effects on proteins, etc. And at the end of the day, you're going to get things like vasoconstriction, platelet aggregation, ACTH release, and also some glycogenolysis. And that's really, I think, what I want to focus on for this lecture in terms of the activation of AVP, the V1 receptor. The V2 receptor activation results in increased cyclic AMP and protein kinase A activity in kidney cells. Ultimately, what that causes is the insertion of those water channel containing vesicles into collecting duct membranes. Okay, and these guys are the aquaporin too for these vesicles that get stuck in the membranes that then allow water to get reabsorbed. Urea permeability is in the renal collecting duct is also stimulated with V2 stimulation, and that helps maximize urine concentration. And then sodium transport in the collecting duct and thick ascending limb is also to a certain extent stimulated, and that's mediated by activation of the sodium, potassium, chloride symporter. So you get a variety of effects with V2 activation. You know, a really big one, at least in my mind, is those water pores and allowing for the reabsorption of water. This slide is from Goodman and Gilman from the old version, shows V2 receptor action. So when the V2 receptor is bound, you get this increase in cyclic AMP production, protein kinase A activity. Proteins get phosphorylated, blah, 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 second messenger system stuff. But a key is these vesicles, these water pores, get stuck into the membrane. They get moved from, they're hanging out in the cytoplasm. Their movement is stimulated into the membrane, where then they'll allow for the passage of water. This just shows it in a different way. So I'm not going to go over this slide too. It's about the same thing. I guess, well, maybe the one thing that makes this slide useful is it actually shows which side things are happening. So these water pores are on the luminal side, right? So they're taking water in from the urine, and it's getting cycled back into the blood. Okay, this slide shows V2 receptor activation and all the things that can happen from that. You know, it's just in word form. So you get V2 receptor activation, rapid insertion of these preformed aquaporin 2 channels in the apical membrane, principal cells in the collecting duct, and you get an increased permeability of the duct to collect water and ultimately conservation of water. Okay, just going to move on to agents affecting vasopressin, vasopressin receptor agonist and antagonist. They're all nonapeptides, so they're all 9 amino acids stuck together. And what they do is they bind V1 or V2 receptors, and they increase or decrease their activation. And as you would assume, they're used to either enhance or decrease the effects of vasopressin. And I needed something to balance that slide out, so that's pretty simple. Vasopressin receptor agonists and antagonists are vasopressin analogs. Funny how that works. Okay, the effects of these agents, pharmacological agents, on vasopressin actions include, once again, there's drugs that increase the antidiuretic response to vasopressin. So these guys are going to alter, to a certain extent, kidney function. So things like NSAIDs, which probably through their effect on prostaglandins, can actually increase the antidiuretic response to vasopressin, so increase the reabsorption of water. So NSAIDs can do that, and so can carbamazepine and chlorpropamide. And drugs that are going to inhibit the antidiuretic actions include lithium, and then demaclocycline. It's an antibiotic that I don't know. The key is that there's different drugs can actually increase the overall effects of vasopressin. And so here we're not necessarily talking about causing the stimulation of vasopressin release or the inhibition of vasopressin release, but altering the level at which vasopressin can affect how much water is being reabsorbed, how much water is being conserved. this slide probably should have been moved up. This slide just shows vasopressin analogs. I wasn't even I got to even lean over here to see it, so I imagine you guys can't even see it up there. So it's kind of a worthless slide, and I don't even know how well you could probably see it printed out in front of you. But this just shows the vasopressin peptides and what they do. So if you want to just walk yourself through these, I'm not going to ask any by name. So, but for your own knowledge, if you'd like that, I think really for our purposes, just know, vasopressin agents are vasopressin analogs. There's not a lot of clinical use that I know of for agents that directly affect vasopressin, that are, for vasopressin analogs. So the diseases that affect the vasopressin system include diabetes insipidus, which is a disease where vasopressin secretion or the response to vasopressin is insufficient. So it's like diabetes, only with vasopressin. And people with diabetes insipidus excrete more than 30 liters of fluid per day, and they're usually excessively thirsty. So it can be a fairly noticeable thing. And what happens in these folks who have diabetes insipidus is they get low levels of functional AVP, which is central DI, renal insensitivity. Sorry, this is really categorizing diabetes insipidus. So if you have low levels of functional AVP, that's central diabetes insipidus. If you have renal insensitivity to vasopressin, that's nephrogenic DI. That's usually caused by vasopressin receptor mutation. And then drugs which inhibit the effects of vasopressin can also cause DI. So things like lithium, which inhibits vasopressin, can cause DI. So it's probably one of the reasons you need to monitor lithium levels and monitor people on lithium. And also, vasopressin can sometimes be broken down too quickly by vasopressinases, and that will cause diabetes insipidus as well. For treatments, basically you use these vasopressin analogs for central DI. So once again, keep it real simple. Vasopressin analogs, they're nonapeptides that are going to, with diabetes insipidus, to treat it, you're going to activate ones that are agonists. Amiloride is used for nephrogenic DI. Remember the diuretic amiloride in that sodium channel that it affects. And what that does is that blocks the uptake of lithium, and, sodium and lithium in the collecting duct. So lithium can't have so many negative effects. Thiazide diuretics, paradoxically, reduce the polyuria in non-lithium-induced nephrogenic diabetes insipidus. So diuretics and DI can clash. Be careful using those. And then indomethacin, has also been tried for nephrogenic DI. That's actually a prostaglandin synthase inhibitor. I don't think it's still being used, but it's at least somebody out there thought of trying that as well. Okay. Another disease, does anybody know how common diabetes insipidus is? I should have got that information. Did Corey happen to say anything about that? No, okay. I'll have to look that up and maybe have that for Friday's lecture. But another disease that affects the vasopressin system is syndrome of inappropriate secretion of antidiuretic hormone, or SIADH. Symptoms of this are hyponatremia, so not enough salt in the serum, and then hypoosmolality. Osmolality goes too low. SIADH involves obviously the inappropriate secretion of vasopressin, and it mainly occurs when high fluid intake is found in addition to inappropriate AVP secretion, and this can cause, as one would expect, well, malignancies, but pulmonary diseases, CNS disease or injury from changes in osmolality can cause problems. Pulmonary disease can cause this. Sorry, I have that backwards. CNS diseases, which are going to affect vasopressin secretion, can also cause it. And surgeries can, for some reason, cause this as well. And the book didn't really go into too much why that was. Okay. This thing can also, probably the key point, can be induced by drugs. So drugs that are going to affect the vasopressin secretion can cause this inappropriate vasopressin release. Things like fluoxetine, haloperidol, tricyclic antidepressants, sulfonylureas, etc. Thiazide diuretics, clonidine, enalapril, methyl-dopa. There's a long list of agents that, through their effects on dopamine or acetylcholine, can, and other things that vasopressin is involved in, they can cause this inappropriate secretion of vasopressin. Okay, so, something to look out for if somebody starts having water regulation problems. You want to figure out what they're taking that might be causing that. Other diseases out there that affect the vasopressin system can include congestive heart failure, where you get this pile up of fluid in the heart, right? Nephrotic syndromes, where there's kidney problems, and both these can actually reduce effective blood volume and mess with normal vasopressin secretion. Hypovolemia, which is exacerbated by excessive diuretic use, can actually cause an over-response, the over-secretion of vasopressin, and that then can result in hyponatremia, so not enough salt. So all fits in with the fluid regulation thing. And that's it. A short lecture today. Any questions? Leave everybody dazed and confused. So, okay. Got questions or whatever, let me know. I think if you read through the slides a few times, it makes sense.